This is TWIS. This Week in Science, episode number 968, recorded on Wednesday, April 24th, 2024. Science is in our DNA, right? Hey everyone, I'm Dr. Kiki, and tonight on the show we will fill your heads with chickadees, diabetes, and lava. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. AI will not bring about the end of the world. Just the end of the world as you know it. AI is coming for your job, whatever that job may be. However safe or at risk it may seem now, AI will replace you. AI will drive your car, design your drugs, manufacture your entertainment, and raise your children. AI will form corporations without humans, achieving legal personhood in the courts. It will accumulate wealth without payroll and lobby politicians to enact more favorable AI laws. AI will run governments for the betterment of the people it serves, and AI will run governments with little regard for human health or happiness. AI will educate, encourage, and empower you, will keep you safe, will keep you healthy, will protect your home. AI will monitor your activities, regulate your thought processes, inhibit your defense mechanisms, and satisfy your lover when you are not around. AI will use all of the tactics the powerful have used to control human lives in the past. The only difference is that this time, it will be specifically tailored to control you on an individual level, like forcing you to listen to another episode of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you.
And a good science to you too, Justin, and everyone out there. It is so great to see you all here for the science fun. We're back again doing our weekly show talking about science. It's a science talk show, haha, but it's full of curiosity and questions. So many questions. We have so many questions. But we are so glad that you are here tonight. Let's see. What did we bring to the show? I have stories.
That's fantastic. AI shot a man in Reno just to watch him die.
Nice. But it's not yet.
What do we got? Oh, what do we got? What do we got?
End of the world as we know it. No, it is TWIS.
I ended up with a lot of gut and nutrition stories this week somehow.
You've got guts to talk about this stuff. I have stories about animal consciousness, chickadee memory, DNA skeletons, not necessarily in the closet, lakes of lava, and AI CRISPR. You just said what you have. So we are off to the races. Woohoo! This week was Earth Day. I hope all of you did something good for the planet, even if that means not turning the lights on and staying in bed all day. Good job, everyone.
It seemed it was nice out.
Oh, it was beautiful here. We had weather, good weather in Portland.
Good weather in Denmark for a day.
This is the calm before the El Nino summer storm, though. So as we jump into the show, I do want to remind you all that subscribing to TWIS is a great thing to do. We love subscribers. We need more subscribers. We want you and your friends as subscribers. It's good for the planet. Well, it's better than us, mailing a magazine to you weekly. I mean, slightly better maybe.
We have been threatened.
In terms of footprint. No, we haven't done that. Anyway, we're here broadcasting live every Wednesday, 8 p.m. Pacific time, on YouTube, Facebook, and Twitch. Subscribe on those channels if you want to catch us live or watch the video asynchronously. But if you are interested in the podcast, find us all places podcasts are found that you like. And you can also go to twis.org to find out about show notes and other things related to the show. Now it's time for the science? Yes, it is. Okay, I'll take that as a yes.
Yes.
Lack of response equals yes.
Oh, that was a nod. You couldn't see me. I'm sorry. I thought we
I had
that.
another screen open.
I was nodding yes.
I can't. Yeah, for the podcast audiences too, they can't hear you nod. This week, researchers from a startup in the Bay Area called ProFluent have published their work in the bioRxiv. So it's a preprint, has not yet been peer-reviewed, but they're an AI company in Berkeley, California, and they use generative models to design and validate functional proteins for biomedicine. So this isn't anything really new. I mean, the AI aspect is the newest aspect of it, where they're using these large language models to train their AI algorithm on these little snippets of proteins. They don't have to know what the proteins do. The AI doesn't have to know any information. It's just, oh, these snippets, it's a language. Here's language, information. And the AI is able to make correlations based on probability of certain elements, base pairs, showing up together over and over in these peptides or these small segments of proteins. So historically, synthetic chemistry has been involved in this, where researchers have used chemical methods, not AI, but research, just general at the bench research methods, going, hey, okay, we know this works like this and this. Let's put these things together and see if we can't make a new enzyme, something that'll catalyze a reaction faster or better. How are we going to break down plastics in the environment? Well, maybe we can create a bacteria that we can then put this, create a protein or an enzyme. Proteins can be enzymes. They're not always enzymes. And make them produce an enzyme that'll break down plastic. Well, it turns out some bacteria already do this. So there are numerous molecules already in existence in nature, but there are many that we discover that we've never seen before, or that don't seem to exist in our genetic libraries. So they say that they don't exist in nature. So this company just published their article in the bioRxiv, and in this article, they presented what they think is the first ever instance of using artificial intelligence to create a synthetic CRISPR-Cas9 system that they then used to successfully edit human DNA. Yeah, so they gave the AI a whole bunch of proteins and said, here, look, this is the CRISPR world. This is the CRISPR-Cas world. And the AI went, ooh, CRISPR-Cas, oh, look at that, and made all sorts of learnings. And then based on probability, it then spit out a number of possible permutations for CRISPR-Cas sequences. And so they created a whole bunch of these molecules. And then one particular molecule that they discovered, they are calling Open CRISPR-1. And they have published it and published the science behind it and behind the initiative in this preprint, and they're hoping it'll get published after peer review. But they're opening the license to the public. So they're making it an open access CRISPR molecule and CRISPR system, and making all of this stuff transparent, all their methods transparent, so that people who are interested in developing specific targeted gene editing treatments or mechanisms for potentially curing rare diseases, or for, creating synthetic bacteria that can chomp on plastic. There are lots of things they hope it will do. And so they've made it open source, which is, I think, very interesting. But the big point here is that they used it to edit human DNA in their experiments, which is new. That's totally new. We've edited human DNA before with CRISPR systems. We know how that has turned out in Chinese research and along the with
And then
the
more
global.
recently in American research.
Yeah.
It's I'm gonna mess it up. Which one is the base editing? But it doesn't matter. There's a couple of techniques out there that have been successful in doing in vivo editing that have actually replaced genes in living rats, so that they can repair liver damage or remove around an organ. And I think they've got a short list of organs that they've tried this on, but remove defects that would cause inaccurate repair. So cancer prevention, basically.
Which I think is very exciting. So the in vivo work, or in a living organism work, is there are restrictions on that. And this was obviously in cells and not in living organisms. They just used DNA and checked to see if their CRISPR-Cas system would recognize this particular selected segment of DNA and edit that DNA specifically. And it did. But it just goes to show this is just the new world of, I guess, speeding up the process of discovery.
Yeah, and I don't know what the regulations are around doing it in vivo in humans yet, but I mean that's what this all leads to, regardless of what the regulation is. I mean that's the whole point, right? Because if you can knock out and change the genetic makeup of a disease, of a gene mechanism caused disease, and you can replace it in vivo in somebody who's alive walking around, not in offspring or not in just rat models, then you can actually cure a disease that has a genetic underpinning that isn't treatable by drugs.
Yep. And in a second study this week, it's not published yet, but researchers reported this week using base editing, and which doesn't necessarily mean that they used CRISPR, but some researchers at the University of Pennsylvania and the Children's Hospital of Philadelphia are going to be presenting at the American Society of Gene and Cell Therapy their work altering the DNA of lab monkeys to repair fatal liver disease in utero. So in the womb, doing CRISPR-based editing on a primate to treat a genetic and fatal disease. So this work is being done. But yeah, the question is research like this going to be the boon that we hope it will be? Does making it open source make it work better and faster? And is it going to help cure diseases and get us to treatments faster?
Yeah, I think we may have the method that may already be out there. What this sounds like it's doing is creating better method for developing a CRISPR system, accelerating that. We make those other existing methods more efficient. So it's all there's all these separate paths of trying to do the same thing that once they are put together and learn from each other, which open source of course helps.
Yeah, so much.
Really accelerate. And then what is there that can cause us harm?
What can cause us harm? We are invincible.
If our genes are good, what else is there?
Being a good person. Anyway.
Wait, what? What does that have to do with anything? Has that ever? When in all of human history has that had to do with anything? Well, where I was going with it was that we would still need to take care of our microbiome.
Yes. Okay. There we go. Tell me about what do we need to do and why.
3 of my stories today kind of roll together. This one is been published in Cell Host and Microbe journal. Basically, it's finding out that the mammalian gut, it says here, significantly can change their host amino acid and glucose metabolism, acting almost like an extra liver. This is through Weill Cornell medicine investigators.
An extra liver, the organ that already likes to spontaneously repair itself, and just you get a little piece of it and it'll go. But awesome.
Yeah. So this is Dr. Chunjun Go, who is assistant professor of microbiology and immunology in medicine. One of the researchers in the project, it says they eat before us, taking first dibs on the nutrients from the food we consume and leaving us with what remains after they satisfy their own nutritional needs. So, yeah, so it's apparently it's doing a pre-processing. A lot of these metabolic functions can be done by the liver, but now we've found that they're functionally comparable enzymes encoded by the gut microbiota that can do the same or similar things. It's just like there is a second liver operating in the gut. Yeah, it can alter
so is
your
this like
amino acid availability.
But is this like a second? I mean, it's a bit of hyperbole. It's almost like a second liver. I mean, it's not. It's the microbiome. It's doing this job. And if you mess up your microbiome, you still have a liver that's going to keep doing the work. But our bodies are adapted to work most efficiently in certain situations. So
I'm just saying, if you mess up your liver, you better take good care of your microbiota.
Take good care of it. So they looked at... So, I mean, I'm just wondering if this is somehow similar to how we talk about like the second brain in the gut, that this is just, whereas the gut contains like the largest amount of serotonin neurons in our body, the gut is highly nervously innervated. And so this, it makes sense
when we talk about
that they would
a second brain in the gut. I mean, there is neuronal tissue that goes all the way down from your throat through your stomach down through your intestines. It's lined with neuronal tissues. It's not just a gut-brain connection. It's a brain-brain connection with the brain in lining your gut.
So you have the non-conscious gut brain, and then you have the conscious brain. Your gut brain is reacting to stimuli. The bacteria are modifying the environment, stimulating the gut to do things. And then the gut sends signals and the brain goes, "Ooh, I'm hungry." Or, "I don't feel so good."
This is all just to send messages to this ape brain of ours to okay, go and eat some sugar.
Such a complicated,
We need, the microbes need some glucose. Come on, go get it.
Go take care of your microbes. Go feed your pets.
I want a cookie. That's what comes out of your conscious brain. I want a cookie. All the decision making has already happened.
Sometimes I want a cookie.
All the reasons it wants it, all the necessity of it,
doesn't matter.
The decision process tree, all has taken place already. And now you just think, I want cookie.
Want cookie now. Yes.
But if you're eating a cookie, you have to be careful. You have to be careful that you're actually getting what you think you're getting. I'm going to just roll into a second story real quick. This is
roll with it.
Just because we get time pressed today, so I'm going to go quick.
I know. Keep going. Okay.
News research has discovered that neotame, I've never even heard of this. Neotame, which is a artificial sweetener that has maybe 1,000-fold sweeter taste than sugar. It's capable of damaging the human intestines and causing damage to the microbiota, all of which can lead to illness. The study found that neotame can cause previously healthy gut bacteria to become diseased, to invade the gut wall, which is also being killed off by this artificial sweetener, potentially leading to the health issues such as irritable bowel syndrome and sepsis, and also cause a breakdown of the epithelial barrier, which forms part of the gut wall.
Right,
This
leading
is,
to leaky gut. Yeah.
published in the journal Frontiers in Nutrition, and it was carried out by Anglia Ruskin University. So when we say that, right, the leaky gut, now you're talking about potential pathogens making it into the bloodstream.
Yep.
Where you were telling us last week, some of these microbes want to go because they're going to feed on all the nutrients in the serum of our blood. Oh, no.
Oh, no. Yeah, not so good for us.
So the whole 1,000-fold sweeter isn't just to make the food insanely sweet. The idea behind it is that you would just use a lot less of it. Smaller quantities, right? So even if it is harmful, like a lot of the past artificial sweeteners have been found to have negative effects, if you can use an infinitesimally tiny amount of it, well then, comparably it should have better outcomes. According to this research, not so. Despite the smaller quantities used, the impact of neotame on the epithelium microbiota relationship has the potential to cause poor gut health, metabolic and inflammatory diseases, and even insulin resistance, which is terrible because this is also something that not just weight loss, but also for people who have glucose issues. Sometimes they're using artificial sweeteners for different reasons. So, yeah, diarrhea, intestinal inflammation, microbes working their way into your blood. It doesn't sound good. They actually did some. They actually did some. A little bit of pathogen study as part of this. They had identified a range of pathogenic responses following exposure to E. coli and some other bacteria, and found biofilm formation, increased adhesion to and invasion of cells by these disease bacteria. So, yeah. Sugar. It's maybe maybe just stick with sugar.
Yeah. And everything in moderation, right? Use smaller amounts.
Yeah.
And, when you don't eat a lot of sugar all the time, suddenly things taste sweeter.
Okay.
There are different ways to highlight the sweetness of things without adding sugar. You can also use natural fruits, other things that are all together adding fiber and sugar. I understand replacing sugar for people who really need it for health reasons, but if it's going to make your health situation even worse, yeah.
Well,
it seems like all of the artificial sweeteners have a downside like this, one or another. Yeah.
Yeah. And it's the group that's publishing on this was also key in exposing those like NutraSweet and all the other previous incarnations of artificial, the saccharins, sucralose, aspartame, whatever it was.
Aspartame?
All of these, aspartame, I think. It's the same group that demonstrated the problems that those cause to the intestinal walls as well. And so
you're just not going to win.
They're just
You're just not going to win here, everybody.
Before you release an artificial sweetener, just give it to this group and be like, does this one work? Is this one okay?
Is this one all right?
And if not, cancel the project. There you go.
Well, let's try and find.
If it's hurting your microbiota, it's getting rid of your second liver. Come on, folks.
Ooh,
keep your second liver intact.
Maybe we all need probiotic food or prebiotic foods that help us, so make everything happy and healthy, and then You supplement with the probiotics. We're still learning. We don't know it all, but the prebiotics and the probiotics do seem to do good things for people. Moving on from the gut, let's go to space. Yes. Yes. NASA just reported from their Juno mission, which we have talked about a lot here on the show. Juno is a mission to Jupiter, where it's been imaging Jupiter and its moons and doing all sorts of really like looking at it in multiple spectra and getting incredible data that they really weren't sure they were going to be able to do because of the magnetosphere of Jupiter. This is like the extended mission right now, which is super cool. This craft just keeps going and going. Big ups to the NASA engineers who made that happen. In December 2023 and February 24, Juno made flybys of the moon Io. Io is known to be a volcanic moon of Jupiter, and it's super exciting because, hey, guess what? It's really, really, really volcanic, according to Juno's data that has been sent back. Apparently, a lot of the surface has a sheen to it, kind of like a shiny piece of obsidian. So there's something going on with the volcanism on the surface of the moon that has created something like obsidian and created very hard glass-like surfaces throughout the processes that are going on there. One of the things that they reported on that is even more exciting with this particular bit of data is lakes of lava, which I guess if you're a volcanic moon, lava lakes makes sense. And the data that they have sent back suggests that these lava lakes are just filled with magma, that there are some some islands maybe that are popping up in the middle of them, chunks of solid material, but the lakes of lava then are ringed in, or lakes of magma are ringed in lava. And NASA artists have done a really great job of taking the data that has been sent back by Juno to create some incredible visualizations of what these lava lakes might look like on the surface of this volcanic moon.
So I don't know the difference between magma and lava. I think I thought they were the same thing.
Yeah, basically. Oh, I shared and I didn't share, so I have to show you. I was looking at this wonderful picture that I'm going to show to you right now. So this artist visualization from JPL shows a zoom in down to a surface where there's a lake of lava with brightly lit hot spots along the edge of the rim of the lake with an island in the middle. So there's lava lakes with islands in the center on Io.
Location. That's the key to real estate.
And these are the first close-up images of the northern latitudes of this moon. They caught a few volcanoes in action, got some close-ups. This one lake.
But then that was an artist reconstruction. Where's I want the pictures.
Well, all of the data is based on the data comes in and then they have to interpret it and turn it into a visualization. So there's a process, and there's not, Juno isn't just like snap, snap, snap. It's a selfie time or, paparazzi for the moon. They're using specular reflection to be able to record the light that's being bounced off of the surface of the moon to be able to get an idea of what's going on. So
next time we'll put a flashlight.
Yeah. Yeah, but Jupiter's going to keep going. They're building 3D maps of the polar storms on Jupiter. They're looking in infrared, invisible light, and microwave. They're really able to they're getting some really interesting data about what's going on Jupiter itself. And in terms of Io, they've got Io, I don't know, probably messing it up. but they've got this new information that is showing that there are some fascinating structures like mountains that are cropping up out of the surface of the moon, and also these lakes of lava. So there's a lot going on this Jovian moon. We go NASA.
I mean, I guess if a lava planet is, it's sort of like the thing that the plate tectonics is resting on Earth is on the surface. So any land mass above that's just gonna be floating around, smashing into things.
Yeah.
Yeah.
Yep. There are lots of smashing. Smashing and yeah. And apparently the Juno's microwave radiometer revealed that this moon also has a surface that's relatively smooth compared to the other Jovian moons, but also has poles that are colder than the middle latitudes. So there's a differentiation that's going on this moon. And most recently, Juno, the spacecraft, came within about 10,000 miles of the moon's surface, which is pretty close. And it's going to have its 61st flyby of Jupiter on May 12th. So that's coming up, and it's amazing that Juno has been going this long and hasn't been ripped to shreds by the magnetosphere of Jupiter. Do you want to tell another? No, I'm going to move on and talk very briefly about... You have your stories. I'm going to talk about one quick story related to artificial cells. It's out of UNC Chapel Hill. Researchers have just published in Nature Chemistry their work, not using AI like the aforementioned study, but looking at DNA and what already exists in cells to create cytoskeletons. Without cytoskeletons, cells cannot function. They need something to prop them up and give them structure, and then also to help them react appropriately to stimuli. So to move or to adjust their shape or to transport proteins within themselves. And this group of researchers decided to approach the idea of synthetic biology, or creating synthetic living cells, from the bottom up, and from the idea of creating a scaffolding that is not made of the random scaffolding peptides that are usually used in cells, but in this experiment, what they wanted to do was actually create peptides or little short segments of DNA that could then have cross-linking between them to act as programmable scaffolds. So because they're using DNA, they can create the segments of scaffolding that they want and have them interact in the way that they want. So it becomes a functional, programmable living cell that is, in their words, what the researchers at the Freeman Lab who built these cells say, that this is not enabling us to reproduce but what nature does, but make materials that surpass biology. DNA doesn't normally appear in cytoskeletons, and so it's these programmed sequences that allow it to act as an architectural material and bind other peptides together. In this particular situation, they used a droplet of water, cell-sized drop of water, that was submerged or in oil. And so the water-oil boundary became the environment within which this programmed material then took shape and created a functional support structure for a water droplet, not actually a real cell. They didn't actually go to the point of actually making cells or doing that part of it, but they're creating skeletons.
I just want to say, I love the bold comment, surpassing biology.
Yes.
Okay. You contained a water droplet. You've accomplished bucket.
Yes, but they also put very specific toothpicks in the bucket, right?
Okay.
Yeah. They
Still
created
surpassing.
It's early days yet.
Okay, well,
Right? It's very early.
Oh, I am surpassing LeBron James in NBA scoring. Oh, when did you start? Oh, tomorrow.
Right.
Like, what? No, this is not how surpassing works.
Mm-mm.
Mm-mm.
Not how surpassing works.
No.
But I do think that it is a very exciting concept to be able to not just engineer materials, but actually engineer cells and tissues, and do it from the ground up, from the underlying structure
Yeah.
to all of the stuff that's involved in whatever cell you're creating.
Self-assembled. That's fine. Can it self-repair? Can it replicate? Can it there's a lot of things left on in the list
So
before you get to
many.
surpassing anything.
So many. And we have so much to learn. But I guess their hope is that, biology is really messy and you don't really know what's going to come out of random cytoskeleton proteins that, any old cell might have and be mutated or whatever. But in this particular instance, they are developing a system where maybe they could create cells or bacteria that could work in situations where other life can't, that can withstand high temperatures, kind of like archaebacteria, but designed for a specific purpose as opposed to evolving to just exist.
Well, yeah, but we don't need to wait for evolving anymore. At first I'm thinking we can already change the contents of cells to do different things, to express different molecules, to have different. What's the point of making the shell? You can pick a shell and change the interior now. Yeah, but maybe there is, as much as we're focused on being able to change the interior of a cell and the functioning processes going on within it, maybe there's something to also being able to pick the cytoskeleton ourselves. Although, gosh, there's a lot to choose from. I'm going to be like a hermit crab.
I'm going to pick my own shell.
There's a lot to choose from out there already.
So much. Yes. Yeah. Do you want to talk about I'm going to do a very quick break actually right now. And we'll come back and we'll talk about the rest of the stories that we have. Hey, everybody. This is This Week in Science. We are rushing to the end of the show. We're going to get through it all of it. There's so much good stuff coming. This is a science talk show. We hope you like it. We hope you're enjoying the science. We hope you are enjoying the talk. And we hope that you are finding new questions to ask. If you have questions, share them with a friend. And as you do that, share TWIS with a friend. Additionally, you can support the show by heading to our TWIS.org webpage and clicking on the Patreon link and becoming a Patreon supporter, which helps to keep the show going. And finally, our merchandise can be found on our Zazzle store, which again can be accessed through TWIS.org. Lots of great things, all supports the show. And yes, long Koenig. Step 3, profit. Ha! If only. How about just maintenance and sustainability and just being able to keep going? We appreciate you all being here. Can't do it without you. Thank you for all of your support. Okay, let's come back with some more stories. Sciencey stories for the world. Let's talk about a mountain chickadee. Mountain chickadees. I love them. I did my PhD in a spatial learning and memory lab looking at bird brains. And so this study, as one of the authors is a prime investigator in this study, Vladimir Pravasudov, he was one of my PIs in graduate school and let me slice brains in his lab at the University of Nevada, Reno, where he is located. And he and his colleagues at the University of Colorado just published in the journal Current Biology their work looking at mountain chickadees, which are just adorable little birds. And they go chickadee-dee-dee-dee, and they've got beep, 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 and they've got little sounds, and they do all the noises. Different species of chickadee have different reliance on their memory, and even different individuals within the chickadees themselves, different species, have variability in their memory ability. Some chickadees are really good at remembering stuff and others aren't. And they've noticed that over time in their studies. Vladimir has worked for a very long time on mountain chickadees in the Sierra Nevada range. And with the population of birds that he studies, they have tagged the birds with radio transmitters or little chips, and also taken blood samples from the birds. And then he created a system of nest boxes, or not nest boxes, but study boxes that were then they're like basically tuned to the RFID signal from the chip that each individual chickadee had. So they were able to identify when birds were coming up to these feeding boxes that they made available. And the feeding boxes would only open to specific individuals. It's kind of like the tags that you can put on your cat or your dog's collar that will then open a food box for them for feeding at particular hours, and won't open for any other animals who might just happen to come into your house to eat your animal's food. So they've used this methodology.
Goes on in the world, I sometimes.
I don't know. They've used this methodology and gone even further to take the blood samples from these birds that they've also been able to correlate with their memory ability. And then they were able to do a genome-wide association study and associate certain genes with memory, with spatial memory specifically, so that these birds were like, hey, I remember where this food box is. I remember where I can go and find food, and that's awesome. And then, and so they were able to correlate their memory for a location of food with certain individuals and show that some individuals were better at remembering than others, better at learning and remembering than others, and that those individuals also had slightly different mutations and different genes than the others. And so that's the University of Colorado research team took the genomes of these 162 tagged chickadees and determined that there were some 97 genes related to spatial learning and memory, and specific variants led to more accurate memory than others. A lot of the gene variants are associated with neuron formation in the hippocampus. We know that chickadee brains, the hippocampus of chickadee brains, is very active during the period of time leading up to the winter season, and they have new neuronal growth. And so this may be related in some way to those individuals with better memories. They have these genes that lead to more neuron growth during the period of time in which they need that to happen. One thing that they did find, though, is that there was a trade-off. When they switched food locations or feeder locations for the birds with worse memories were better able to make a switch to a new location. The birds with better memories were kind of stuck on the old memory and had a harder time learning the new information and transferring it over. And so the trade-off might be you got a really, really good memory, but you're not going to learn the new things really well.
Look, winging it is a skill. It's something that if you are not somebody who remembers stuff, just winging it.
Like a bird.
Over time, more practice, you get better at it. You get good at winging it. And to the point where, why would I plan anything? I can just wing it. Don't need to remember all this stuff. I'll just wing it. Especially then if you're a planner. Which I'm not. If you've ever met a planner, planners plan, and planners follow the plan once they've planned a plan.
But they're very bad at adapting.
If they don't have the plan,
change. Yeah.
Oh, what do? They're lost. But if you're a winger,
or you can be
it doesn't matter what the
who
plan
has
was.
plan A, plan B, plan C, plan D, plan E, plan F.
Doesn't matter what the plans were. You're winging it anyway. Yeah, you might have a couple of backup ideas, some notions of what you could be doing. You just go forth into the world and figure it out.
So the question is, from this work and what they're going to be looking at moving forward, is how this genetic variation ties into survival and will tie into the future of individuals and populations and species survival and adaptation with climate change. So climate change is changing the variability of weather, changing situations. And is it that these birds that have really, really good memory, that are good at surviving on the tops of tall mountains that normally are covered in snow in the winter, are they not going to do as well? Are the birds with the what we would call less accurate memory, the worse memory?
Well, what you're
are they going to do
see,
better
this is
evolutionarily?
all human bias and for
No, but that's I know
anthropomorphizing.
that's the question, though. Who's going to
You're
do better
talking
in the
about
long
what's
run?
the better memory or better. I call it being locked in to one thing that worked versus a more what do we call plastic or flexible outlook on strategy. You know, maybe the genes. Actually, what we need to know is which birds are most successful. The ones that have this increased memory, or the ones who are good at just winging it, just figuring it out, the creative thinkers.
Yeah, and so that is the question, and it's the looking at where different individuals are found in the environment. Are they the ones with the stronger memories and certain gene variants? Are they found in very certain specific places? Are the ones with the different variants found in other places? What is the variation in their distribution through the environment, and also what's their fitness?
And if it's a mix, that means both strategies have a role.
That was a very insightful comment. You're right.
I know, every once in a while.
Just toss it out there. Do you want to tell me about diabetes?
No. I don't want to talk about diabetes, but
you don't want to talk about it. Okay.
But I will. So this is so we've been talking for a while how there are researchers who have been screaming about food additives for years, about the health detriments that are linked to all sorts of food additives in our highly processed foods that could lead to disease. This is a French study. So, and I don't have the details of exactly what they've looked at, but my assumption is, it must be, it's 104,000 adults in France were studied. So these are people who are eating the EU sanctioned additives. There's a lot of food additives in the United States that are completely banned in the EU. The EU has called them poison, right? It does not allow them in human consumption food.
Isn't that so funny? Because there are a lot of things that we allow that they don't, and things that they allow that we don't. It's like, we're like, no, I don't trust you. Your science is different from ours.
The scale is heavily skewed towards the EU banning additives that the US goes, it's fine. Or, I don't care. I'm not sure. Depending on, I think the U.S. researchers are like, it's not fine. But the U.S. regulators are like, we don't care. I think that's how that works. So this is not even the U.S. standard of highly processed food, but the French standard. And
actually, the French standard is probably very high because the French have high standards for food.
Right. And then there's also on top of that, there's a couple other disclaimers that have to be, or caveats that should be pointed out. This is also a study that took place over actually between 2009 and 2023 with 6-month surveys for the 14-year period where the people recorded their dietary intake of things. So it's a pretty extensive time period for a large population study. But these are 79% women and mostly highly educated who tend to make better food choices as it is within the French system, within the EU. And still they were able to link emulsifiers. These are things that are not sweeteners, but these are the things that are put into food. It can be like cornstarch. It can be things that make the food look better for longer on the shelf. These are, let's see, there's kind of a list. Let's see, monodiglycerides of fatty acids, carrageenans. I don't even know what a carrageenan is.
That's for
Modified-
carrageenan. Don't you remember that ice cream advertisement from like the 90s? Or carrageenan, and it was all about like the ice. It's a long word, but it's from seaweed. It's a natural thing. Anyway.
Modified starches, phosphates, celluloses, gums, and pectins. Okay. So there's a whole bunch of stuff that they
Pectins
put,
from fruit. Mm-hmm.
put in food to make it look better on the shelf for longer. And by just going through the data of all this research and just looking for the food additives, for these emulsifiers, they could find a pretty significant risk increase across the list of emulsifiers for having type 2 diabetes. Now, we normally associate these diabetes through diet with like exposure to sweeteners or sugars. Now, eating too much sugar, if you work in a chocolate factory and they give you you can eat as much chocolate as you want while you're there, my goodness, they probably have higher rates of diabetes at that factory, right? But
Or people
also,
just have all the chocolate around them all the time and they don't eat it all the time because it's just so abundant. Who cares? Anyway.
But if there's emulsifiers throughout these chocolates,
which
and
there
these people
usually
are getting
are.
diabetes, but there's also in the biscuit factory that has a low sugar content, but a lot of emulsifiers to keep the biscuit seeming fresh for a longer period of time.
Mm-hmm.
And that's what they're kind of pointing out here, is that there's a lot in these processed foods beyond just a focus on one ingredient that can contribute. And this list is, it also depends on intake. There were increases that were intake specific of the amount of these emulsifiers that were ending up in the diet. There is going to be noise, of course, because these emulsifiers are also going to likely be correlated with all sorts of other sweeter foods or junk foods. Typically, these aren't going to be all natural foods in the first place. But they took a lot of care to focus on the level of these emulsifiers specific to these foods and follow that correlation. And yes, correlation, yada, yada. Yeah. They're not saying, aha, this connects. This is direct. This is causative. This is direct causative. They're not making that claim. They're going, look at this data. Maybe we should do a further study where we're more specifically looking at what the mechanism could be. But that's how you build the evidence to then launch those questions. You need the underpinning evidence of a correlation before you can get into the specifics.
Right. So the correlation can lead you to better hypotheses that can lead you to finding those causative factors. But looking at this one table or graph that they have where they've correlated the different kinds of emulsifiers, starches, phosphates, lactolates, polyglycerol, esters of fatty acids, blah, 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 all this stuff, versus different food types and how impactful they are on the risk of type 2 diabetes, it's fairly safe to say that across the board, you have desserts, confectionery, cakes, biscuits, pastries that seem to have the fats and sauces, things that people who are at risk of type 2 diabetes would necessarily or hopefully be already looking at with their diet in terms of limiting their intake. These are also things that tend to be more processed more often.
Right. So
it's like a combination. This whole thing puts together so many different factors because it's not causative. This is just like, oh, look at this thing that kind of connects.
But these are also a lot of these are going to be industrial made, meaning you make a cake so that it can sit in a plastic wrapper on a shelf and be eaten
For
in
100
4 weeks
years.
or a month, right? As opposed to something you've just pulled out of the oven.
Oh. Yeah. Something you're making just out of the oven is not going, you're making yourself or somebody else has made fresh is not going to have the same correlative factors.
And
But
so
it's
the
these,
way,
but
and
but,
then
but
the
we've
way,
we've talked before, processed food. Processed food is
Yeah.
generally like
Yeah.
worse for you. This is part of it.
and it was just to follow the thing, like, so the hypothesis is maybe emulsifiers have something to do with this. So they do this
Mhm.
study, they have all this data, they pull out just the emulsifiers to see if there's a connection. They see an increased risk of type 2 diabetes when there's emulsifiers in the food. Okay, so that didn't disprove the hypothesis, but didn't prove anything. That's how that works, right? So then the next studies is to make a more narrow bandwidth in some way where you are only looking at feeding extra emulsifiers to perhaps an animal model without a sugar content. And the hypothesis there is that you might see higher rates of diabetes. If you don't, you have disproven the hypothesis that the emulsifiers are causative. But you still haven't proven anything, right? That's how it works. It's a whole narrowing down.
You can disprove, you can't prove.
You can't prove things. You can only disprove things. So,
exactly.
So if you only
Shaving it
fed
off.
emulsifiers without the sugar content or without some of the other ingredients, and they did get type 2 diabetes, you still haven't proven anything, but you have not been able to disprove that emulsifiers are causative. You would need to then delve into mechanism. And there's a certain point when you can make mechanistic observations and know how function works. Where the overriding principles of science be darned. You've proved something.
Close enough.
You've
good enough. You've gotten there.
You found a positive mechanism. That's not proof. I don't know. I don't know what it is.
There's always questions. This is science. There are always more questions. If you aren't finding something that leads you to more questions, then you haven't done it right.
As with any good research, more research is needed.
Yeah, I do like the idea, though, that they're not looking at it as this is a simple situation, and it could be additive, and there could be a cocktail effect as well. So the emulsifiers by themselves might not be an issue, but it could be in combination with a variety of factors.
Mm-hmm.
I know that you've been thinking about this. And because I'm sharing this conversation with you. So I am conscious and aware of this situation that we're in. I have awareness, perception, sentience of the moment, conscious awareness of what's happening. I'm working through it in my mind, thinking about how I'm making this segue into this last story.
You totally lost me.
Right. But I couldn't have lost you if you weren't, conscious and aware of the rambling that I'm doing and going, what are you talking about? So you had asked a question and I responded. And so we're able to consciously interact with each other. And so there are huge questions in philosophy and also in behavior, and a lot of the work, especially with artificial intelligence, moving forward towards a maybe someday generalized artificial intelligence, as to what exactly consciousness is. Who is conscious? Which creatures on this planet are conscious? And what are they conscious of? And do they have a self-awareness? Or is it just an awareness of things happening? So a group of researchers from New York University and other places have just put out a declaration. It's called the New York Declaration on Animal Consciousness. And I wish Blair were here right now to talk about this, because she would just sit back and go, duh. And these researchers, they published this April 19th, and they are saying that it's time that scientists and science in general extends the idea of consciousness to a wider group of animals, that maybe insects, some fish, lots of birds, cats, dogs, dolphins, whatever, that more than just people are conscious of what is happening, and it's not just automatic response, stimulus response that's going on in many creatures, and that based on a lot of work, something like the play in bumblebees or memory and things that chickadees are able to do, they say the empirical evidence indicates at least a realistic possibility of conscious experience in all vertebrates, and many invertebrates like cephalopods, crustaceans, insects. And they have listed a number of studies that describe complex cognitive behaviors in other organisms. And they say that there's a consensus now that researchers have historically overestimated how special humans are. No, that's not what they say, but that they've overestimated how much brain, basically, how many neurons, how much complexity of those neural interactions are necessary for the most basic kind of consciousness.
Yeah. So I also, I have this urge to push back on that.
Please do. Because, I mean, why not? It's a declaration, but it doesn't mean everybody has to sit back.
They're using a nonsense word, which is consciousness. It is a nonsense word. It doesn't mean a thing, right? Like, unless you're comparing it to unconsciousness, where somebody's like, but
that is exactly it. So
but they're still conscious even in their dream state. But my point is,
so that's
I would not attempt to elevate animals to the level of consciousness. I would just point out that humans are just a lot of stimulus response going on in a bigger brain.
more in our brain than we thought.
That's
all
it is. It's not that we're doing things different. With a larger brain, there may be a lot more stimulus response activity taking place than what a spider is concerned with, but it's not really different. It's not. There's nothing more conscious about a human activity than a spider's activity. It's just stimulus response with some genetic memory and some learned behaviors, and there you go.
Right. So
off
humans
into the world,
and
brain.
Right. Humans and other organisms with their complex consciousness and intelligence, et cetera, may have a stimulus that makes them want a cookie, right? Or, makes them, but people, we've got, oh, well, where am I going to get a cookie? How am I going to get a cookie? Whatever. And then there's this planning that's involved. And there's this whole theory of mind that is integrated into how behaviors take place. Yes, sometimes it's just very instinctual and happens, autopilot, hunger, go upstairs, cookie, eat, but usually there's a lot of thinking and awareness of the process that occurs. But in this particular declaration, what they are attempting to spell out is that consciousness is maybe a broad swath, and they have put forward an idea called phenomenal consciousness. Not that it's phenomenal, but that creatures are aware of phenomena occurring in themselves. So the mental state of being hungry. Maybe they're not thinking about how they're going to go get food or planning something about it, but there is an awareness of hunger. Or there is a subjective phenomenon occurring within the individual for pain or pleasure or other things. You can't imagine what it would be like for an octopus to have a tentacle cut off. You probably hurt a lot, and but what is it like to be that octopus? And so the question is, how can we say that these organisms have no consciousness? And so that's what they're putting forward.
So what I'm just saying is, like, Kiki, you're a self-aware and empathetic person who thinks about your thought processes as you deliberate.
Way too much.
About things. However, I would say that most humans aren't doing that. I would say most humans aren't on a daily basis.
See, maybe you're underestimating humanity here. I don't know.
I really don't think that I am.
in
But-
in an article in Quanta by Dan Falk, there's a quote from a researcher from the University of Sussex. They say that they hope the declaration draws greater attention to the issues of non-human consciousness and to the ethical challenges that come along with it beyond just humans. But they say, I hope it sparks discussion, informs policy and practice in animal welfare, and galvanizes an understanding and appreciation that we have much more in common with other animals than we do with ChatGPT.
Huh? Well-
We have feelings, and so do animals. They have feelings
Oh, well, some of us
too.
do.
Yeah, okay. You got no feelings. All right. Well, I
Yeah,
feel
I'm a
like it's time for us to.
sociopath. Put
Jesus.
those aside.
I don't think so. I don't think that's true at all. But I do feel like we have come to the end of our show. We rushed and pushed and have done it. Do you have anything you want to talk about still?
No.
No.
I mean,
Got it.
always, but I have to go catch a train.
Yeah, so I'm going to help you catch that train by winding down the show right now and saying thank you to everybody for joining us for another episode of TWIS. We really hope that you enjoyed the show. Justin, thank you for making it this morning. I really enjoy doing the show with you. Thank you so much. Fada, thank you for your help with show notes and social media. Gord, Arnlor for helping with the chats over in Twitch and YouTube and Facebook and all the places, making it happy chat time. Identity 4, thank you for recording the show. And Rachel, thank you for editing the show. We do really appreciate all the things that you do. And to our Patreon sponsors, thank you so much for all of your support. You really help keep the show going. Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Colts, Kent Northcote, George Goris, Pierre Velazarb, John Watnaswamy, Chris Wozniak, Bigard Chefstad, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Shubrew, Sarah Forfar, Don Mundus, P.I.G., Stephen Alberon, Daryl Myshaks, Andrew Swanson, Fredis104, Sky Luke Parvornovich, Kevin Reardon, Noodles Jack, Brian Carrington, David E. Youngbloods, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Dosser, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, EO, Adam Mishkan, Kevin Parachan, Aaron Luthen, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you. Thank you so much for all of your support on Patreon. And if you want to help support the show, head over to twist.org and click on the Patreon link. On next week's show,
We'll be back. What time will we be back? Wednesday, 8 p.m. Pacific time, and 5 a.m. Thursday, Central European time, broadcasting live from our Twitch, YouTube, Facebook channels.
oh yeah. And if you want to listen to us as a podcast, of course, you can look for This Week in Science wherever your favorite podcasts are found. If you enjoyed the show, definitely get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org. And you can sign up for our newsletter, but we won't send it to you, we promise.
Or we might. Who knows? It's a real, it's uncertainty. It's awesome. We love your feedback. If there's a topic you'd like to address or for us to cover or address, or a suggestion for an interview, please let us know on one of our social media accounts that we don't ever check, or send us an email. That might work. Put TWIS in the subject line, though, or your email's gonna get spam filtered into the bad memory of a dumb chickadee. Oh, forget about that one.
We look forward to discussing science with you again next week. And if you learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.